Thank you so much. Thank you, Marcus, for the invitation. So as Marcus was saying, a lot of my research in the lab really focuses on the underlying neurobiological processes associated with nicotine dependence. So what I'm going to tell you about today are two of the imaging studies, and specifically our fMRI studies, that focus on some of the components that play a role in why it's so difficult for smokers to quit and to stay quit. And I think as a field, when we're talking about nicotine dependence, we've done an excellent job of informing the population that smoking is bad for them. Everyone gets it. Everyone knows that smoking leads to health-related illness and to actually premature death. But despite this, most smokers have a very, very hard time maintaining abstinence. In fact, even though most smokers want to quit and they try to quit, even when they're aided with a smoking cessation aid, such as nicotine replacement therapy, only a very, very small percentage of them are able to maintain this long-term abstinence. So our problem isn't now really understanding that smoking is bad, but our problem is really understanding why do people continue to smoke despite all of these negative health impacts. So with this in mind, this is the main reason why we've been focusing on using neuroimaging to study nicotine dependence, because we want to understand what's going on in the brain that might make it so difficult to maintain abstinence. And today I'm going to tell you about two of the studies we've done. The first one is going to focus on cue-reactivity, but specifically I'm going to talk to you about the cognitive processing underlying cue-reactivity. And I'll talk about working memory specifically, working memory really being the process of holding information online for a short period of time so you can manipulate it. Kind of like the process that you would use if someone gave you their phone number and you had to hold that information online in order to call them immediately. Okay? And then the next study I'll tell you about is focused on subjective craving. And subjective craving is just that feeling, the desire to go out and smoke. But in this case, I'll be talking to you about functional connectivity, which allows us to look at how brain regions are interacting with one another instead of really showing us which brain regions are active to attack. So we'll be looking specifically at whether brain regions communicate differently as craving increases. So you're going to be healthy and you're going to go for a walk. But on this walk, you came across your favorite bakery. And now all of a sudden you're inundated with cues of something imperative, something you really, really like, which is donuts. And now the problem is it's got you thinking about how much you really like donuts. So instead of continuing on your nice, healthy morning walk, what happens is you might shift your behavior away from what it was that you had intended to do, which is be healthy and go for a walk. Instead, you find yourself going into that bakery and buying and eating a donut. And the same process happens for people with nicotine dependence, such that even if smokers are trying to quit and stay abstinent, even if their goal is to be healthy, coming into contact with smoking cues in the environment, maybe it's seeing their friends smoke, can actually induce feelings of craving and potentially lead to relapse. And the two studies that I'm going to talk about today touch on both of those topics, but kind of individually. So I'll be talking about cue-reactivity in one study and the subjective feelings of craving in the other. So one of the ways that I and many others have actually studied cue-reactivity is using functional magnetic resonance imaging, which uses a gigantic magnet, which allows us to indirectly measure brain activation. And what we do is we have folks come into the scanner, and what I'm showing you here is a picture of our 3T scanner at McLean Hospital. And then while people in the scanner, we show them pictures of smoking images and neutral images. And with this, we're able to identify, well, which brain regions are involved in cue-reactivity. But, one thing that I've always wondered is what are those brain regions doing? So when someone is just passively looking at smoking cues, what does that mean? How is the brain actually processing these cues? In part because one of the earliest studies of cue-reactivity, which actually focused on cocaine craving instead of nicotine dependence, was done by Steve Grant and colleagues. And what they found was that when people were shown pictures of drug-related cues, specifically when cocaine users were shown cocaine cues, there was actually a lot of activation in memory circuits. So this kind of got us thinking, well, how are people processing the memory for smoking cues? Specifically, how is working memory playing a role in cue-reactivity? How is the process of holding information online about smoking cues? How is that going? And is there a difference between how people are processing smoking cues and neutral cues? And so that was the main question that we had when we came up with this next experiment. So specifically, we want to see if the cognitive processing of these stimuli are different. And we did this using a standard working memory procedure known as the delay match to sample task. And it's a pretty easy task. And it's pretty easy in part because we want everyone to perform it perfectly. We don't want to see any behavioral differences between smoking cues and neutral cues. What we want to see is brain differences. What we want to know is what is the difference in brain activation when someone can do a task for a smoking image versus when they can do a task for a neutral image, which allows us to focus on brain function instead of behavior. And what we do in this task is we show people an image for about 2 seconds, and we ask them to remember it across this 10-second delay. And then we show them a test image where we ask them, does this image match able to look at brain activation for memory maintenance when they're asked to hold that information online for a short period of time. And then we're asked during the second image, when we ask them, does this image match or not match the one you saw before? This allows us to tap into memory retrieval. So now we're able to really look at the different phases of working memory for both cue types. And we did this study in about 18 nicotine-dependent people. They were all fairly young, around 25 years old, and we did this to remove any effect of age-related cognitive decline. So we're going to be looking at brain function associated with cognition. We want to remove any other variables. They were fairly educated and also fairly nicotine-dependent, as shown by our FTND score of 6. Everyone smoked about 15 cigarettes per day, and they had about 6.4 pack years of experience. And finally, their carbon monoxide level was about 25 parts per million. And this really shows that they had all smoked recently, and we know, in fact, they did smoke recently because we all asked them to smoke one of their own cigarettes right before they went in the scanner. And this was to standardize the level of nicotine and standardize the level, the recency of smoking before we did any of these other tasks. So when we're going to look at the data, at the results, what I'm going to show you are these 3 orthogonal slices of the human brain. So the first image is a sagittal image of the brain. And so the front is over on the right. The next image is a coronal image, so it's kind of slicing the head down the middle and looking kind of face on. And the last one is a horizontal image, kind of like if you took a slice off the top of the head and looked down. And what I'm showing you here with those orange blobs on top is which brain regions are more active when smokers are encoding a smoking memory versus when they're encoding a neutral memory. The one thing that was kind of interesting is that actually no brain regions were active or relatively more active when people were encoding a neutral memory relative to a smoking memory. What we get is a lot of activation when people are encoding smoking memories. Now, what does this mean? Well, one thing that I thought was kind of interesting is I've seen this pattern before. So what I'm showing you again is the results of our smoking cue encoding versus neutral cue encoding. And this pattern looks a lot like what I see when we just passively show smokers cues. So this is from another study we did of an independent sample of 20 participants. We get the same pattern of activation. This pattern of activation is actually consistent across studies of cue-reactivity, such that this is the result of a meta-analysis across 11 studies. And the other thing is that this pattern of activation actually overlaps quite a bit with the default mode network, which is a network of brain regions that has been starting to be studied quite a lot now. And what people think that this network is involved in is autobiographical memory and self-referential processing. Now, a group up at Dartmouth has been focused on these brain regions in this network and has actually found that the self-referential processing and actually the activation of these brain regions during memory encoding facilitates memory encoding. So one of the things we started to think was that the activation of these self-referential associated brain regions might underlie what sort of processes smokers were using when they were encoding a smoking-related image, which makes sense, right? Smoking cues are personally relevant to smokers. So what I'm suggesting by this, by looking at this pattern, is that smokers actually may encode smoking memories a little bit differently and a little bit better because they're using these self-referential processes. And this is supported by the next piece of data, which I'm going to show you, which is the maintenance phase. So this is looking at brain activation when I'm asking folks to hold that information across the 10-second delay because they know they're going to have to tell me an answer in a couple seconds. Again, we're looking at the 3 orthogonal slices of the human brain, but in this case what we're seeing is a lot more activation for the maintenance of a neutral stimulus versus a smoking cue. And interestingly enough, the activation that we're seeing is in the ventrolateral prefrontal cortex, which very traditionally is one of the brain regions that comes online during this delay match to sample task, because the ventrolateral prefrontal cortex is involved in maintaining information across the delay. So it's nice and makes quite a lot of sense that it's coming online when smokers are trying to maintain information across delay. Interestingly, though, they need more activation when they're maintaining a neutral cue versus a smoking cue, really kind of suggesting that they might need more cognitive resources in order to maintain a neutral memory relative to a smoking memory. Once again, possibly being because they encoded the smoking memory a little bit differently. All right. So the last piece of data from this study was looking at memory retrieval, and specifically when folks were seeing an image that they saw previously, and they were answering, yes, I saw this. And when we looked at this, again comparing smoking versus neutral, we saw a lot more brain activation during the recall or the retrieval of a smoking-related memory. We didn't see any activation during the retrieval of a neutral-related memory. Now, this pattern of brain activation, again, was kind of interesting to me because of where it's located. And it's located in the insular cortex and actually adjacent regions. And the insular cortex has been heavily implicated in nicotine dependence. In fact, one of our earlier studies showed that smokers who had the greatest insular reactivity to smoking cues, they were the ones most likely to relapse later on, sort of suggesting that the insula is playing a fairly big role in cue-reactivity and the way that smokers are processing smoking cues. In work by Nasser Naqvi in 2007, he showed that people who had a stroke which damaged the insula, these folks spontaneously quit. And again, really implicating the insula in nicotine dependence. All right, well that's great, but what does it do? So there's a lot of information about the insula, a lot of discussion about what it might be doing, and one prevailing thought is that it's involved in interoceptive awareness. Well, what that means is kind of your awareness of bodily states. So how you all are feeling right now, maybe a little hot, maybe a little hungry, maybe a little craving for nicotine, sort of the awareness of integrating those internal bodily sensations. And what the insula is thought to do when it comes to cognitive processing is it might play a role in linking those internal bodily sensations with external task goals. And in fact, a memory study actually showed that people who had an easier time of identifying those internal states or those internal feelings had an easier time remembering previously viewed affective images. Which makes sense again, right? So if you saw an image of something that you found emotionally arousing, whether it may be very, very happy or very, very sad or very, very scared, it makes sense kind of evolutionarily that you would want to remember it and remember it easily. So this kind of fits in with nicotine dependence, right? Here are these cues that smokers find very rewarding. It makes sense that potentially the memory for them and the ability to retrieve these memories might be facilitated. So just to conclude this part of the talk, what I've really tried to show you is that the underlying cognitive processing or the underlying neurobiological substrates associated with memory processing for smoking versus neutral cues is different. And specifically that smokers might actually use self-referential processing based on that brain pattern that I showed you to encode smoking memories. Now, this self-referential encoding may actually help them maintain this memory over delay because as I showed you, those neutral cues actually needed more brain activation in order to maintain a memory successfully. And finally, that the insula is involved in the retrieval of a smoking-related memory, which fits into what the insula might be doing in nicotine dependence and shows us that the insula is not only involved and not only necessary to maintain smoking behavior, but might play a role in the retrieval of smoking-related memories. So I'm going to switch gears a little bit, and I'm going to tell you about a different study we did looking at functional connectivity. So again, we were using fMRI, but in this case, we were not actually using a task. We had people go into the scanner and did absolutely nothing. So they were just laying there at rest. And this allows us to then assess not which brain regions are active during a task, but how brain regions are communicating with each other. And one of the reasons why we did this is because we wanted to know really what is involved in the processing of craving. How is the brain involved in craving? And well, a lot of studies have tried to do this, and they've actually implicated many, many different brain regions. Those involved in sensory processing, prefrontal cortex, striatal regions. And so we've got this smattering of brain reactivity, and people trying to say, oh, it's one brain region, oh, it's another brain region. Whereas I actually came across this nice quote by E.D. London, where it says it's tempting to speculate that activation of cortical regions with sensory and limbic functions, along with activation of the orbital frontal cortex, reflects an interplay of related networks. So this kind of makes sense, right? It might not be one brain region that's the craving region, but what it might be is a bunch of different brain regions that are sharing information across one another in order to have this feeling of craving. So we assess this interplay of different brain regions using functional connectivity. Now, as I mentioned, we had folks go into the scanner and they did absolutely nothing but lay there. And what we did is we looked at the temporal correlations of the fMRI signal. So when people go into the scanner, we're measuring a signal, and over time it can fluctuate, kind of up and down and up and down. And different brain regions have different patterns of this fluctuation. And brain regions that kind of fluctuate together, go up and down together, such that their fMRI signal is temporally correlated. These brain regions are thought to be functionally connected and functionally working together. And this sort of thought has really led to a boom in the study of brain networks. And so what we did is we used a technique known as independent components analysis, which is a data-driven approach. So we have no a priori hypothesis. We don't have to identify brain regions. What we're doing is looking for these independent patterns of correlated brain activation. And what we did is we identified an orbital and medial prefrontal cortex network. So we found this network of brain areas. And we wanted to know now, well, does this network of brain areas change as a function of craving? Well, what I'm showing you here again are some horizontal slices of the human brain. And our network is shown in green. So this is the network that I'm going to be talking about. And we picked this network in part because these are a lot of dopamine terminal areas. So these areas have been associated with reward and drug addiction. And in fact, the orbital frontal cortex is thought to be a multimodal integration area leading to hedonic experience. And Randy Buckner also suggested that this area might act as a hub of integration. So not only are these brain regions and is this network involved in reward and drug-related stimuli and drug-related processing, but it's thought that these brain regions might actually integrate information in order to process these hedonic states. So it kind of makes sense that we're going to focus on this brain region and ask how the integration of information between this brain network and other brain regions might change as a function of craving. So in this study, we had 17 nicotine-dependent folks. Their demographics were pretty similar to the other cohort I showed you. Once again, we focused on young smokers, trying to get a stable brain activation without any age-related effects. Again, they were fairly nicotine-dependent, smoked about 15 cigarettes a day, and had about 7 years of experience with smoking, and their carbon monoxide level was about 28. So when folks came into the study, what we had them do was everyone smoked one of their own cigarettes. And again, this was to standardize the time of all of our subsequent measurements relative to the time they smoked. We then did our craving assessment, which was the questionnaire of smoking urges by Steve Tiffany. After this, we had folks go into the scanner at McLean, and we actually did 2 resting state scans, and we did another craving assessment. And what we're able to do now is look at how craving changes over time, and specifically relate how this change in craving relates to a change in functional connectivity. And the reason why we're able to do this is because our scan protocol had 2 different fMRI scans in it. We had one 6-minute resting state scan at the beginning, and then about an hour later we had a second resting state scan, which really allowed us to compare what's going on in the first scan versus the second scan, and then see how this brain network changed, but also changed in relationship to craving. And if any of you were interested, there was in fact a significant increase in craving when we compare before scanning to after scanning. And like I said, what we want to know is if this significant increase goes along with a change in brain activity. So just to remind you, here's our network again. And when we just did a simple t-test, comparing this resting state network at the end versus the beginning, we saw no difference. It didn't change at all, which sort of suggests that this network of activation, this network, doesn't change on average over time. But when we compared this change to craving, we did see an effect. And what I'm showing you here are, again, the three orthogonal slices of the human brain, with the orange blobs now being which brain regions our network is more connected to as craving increases. Talk more with, or more with, or has more temporally correlated fMRI signals with, all of those blobs that are shown in orange. And those blobs in orange are overlapping with brain regions involved in sensory processing, other parts of medial prefrontal cortex, and also more parts of the striatum. So all brain regions that have actually been implicated in craving and attention to smoking cues and cue-reactivity in the past, really suggesting that craving may not necessarily be due to the activation of a single region, but in part is due to the integration of information across these regions. So to conclude that part, this slide says pretty much exactly what I just said, which is that craving may in part be due to the integration of information across all of these images, kind of fitting in with that quote that I showed you at the beginning by E.D. London, and that a significant change in subjective state may also be involved in changes in functional connectivity. I mean, basically the results of this experiment really got me wondering if it's just craving. And I doubt it. I mean, so if there's ever any change in someone's subjective state, be it an increase of negative mood or an increase of craving, does this really have to do with how different brain regions end up communicating with each other? So to conclude everything I've talked about, hopefully I've convinced you at the beginning that smoking cues are in fact important, and craving is important for why we should study these things in nicotine dependence. And that also fMRI may help us understand these neurobiological processes that underlie cue-reactivity and craving. And also that memory processing is different for smoking cues and neutral cues. But specifically, these different processes, these different brain regions that become active, might actually explain that smokers may be integrating this working memory a little bit differently for smoking cues. Specifically, they might be encoding smoking stuff in a very self-referential way, which allows them to more easily hold that information online over a delay period because they don't need to activate as much brain activation during that delay. And they're actually retrieving this information along with using the insular cortex, suggesting that some of these internal states may be playing a role in the retrieval of these smoking-related memories. And finally, that craving may be due to brain regions communicating with one another. So what's next? I mean, I think this talk really focused on a lot of very basic, neurobiology and basic psychological concepts. But, when we're talking about disease states like nicotine dependence, we want to know, well, how does this make a difference in someone's life? What does this mean kind of clinically? So we want to know is, does this memory processing for smoking cues impact behavior? If we're able to disrupt memory processing for smoking cues, can we disrupt smoking behavior? And does this enhanced memory processing really play a role in why it is smokers have such a hard time ignoring smoking cues in their environment? Because you got to think, if someone is encoding memory a little bit more efficiently and they're retrieving it, with their internal states, well, that might be a bit problematic. And finally, with any correlation analysis, you always want to know, well, what's the cause and what's the effect? Which comes online first when we're talking about the relationship between craving and this integration of brain activation. So with that, I would like to thank everyone who's been involved in this study. And as I was telling Marcus earlier, I actually forgot to include my husband on this, who is a cognitive neuroscientist, and he actually played a big role along with Chantal Stern at BU and helping us come up with that working memory paradigm, because that was a task that they typically use in their lab to study very, very basic memory, and we moved it over into imaging of nicotine dependence. And I'd also like to thank NIDA for their funding of this research. And thank you all for your time. And thank you all.